Newly formed or naive B lymphocytes, or B cells, originate in bone marrow from hematopoietic stem cells, or HSCs. A series of progressively more differentiated progenitor stages leads to the production of common lymphoid progenitors, CLPs, which can give rise to B cells, T cells, or innate lymphoid cells. Progenitor cells destined to become T cells migrate to the thymus where they complete their maturation. The majority of CLPs that remain in the bone marrow enter the B cell development pathway. At various points in their development, the precursors of B cells must interact with stromal cells expressing particular proteins, such as CXCL12, that induce the developing cells to differentiate and move in an orderly progression from location to location within the bone marrow. The cells pass through progressively more committed lymphoid progenitor cell stages until reaching the pro-B cell stage. At this stage, the precursor cell is irreversibly committed to the B cell lineage, and the recombination of the immunoglobulin genes begins. The rearrangements of the immunoglobulin receptor heavy and light chain gene segments form the B cell receptor, or BCR, for antigen, determining its specificity. Immunoglobulin gene rearrangements begin with heavy chain D to JH gene segment rearrangement, followed by the stitching together of the VH and DJH segments to allow synthesis of an intact heavy chain. If the first heavy chain gene rearrangement does not result in expression of a heavy chain and pre-BCR, VDJ rearrangement on the other chromosome is attempted. This chain initially pairs with a surrogate light chain, allowing the cell surface expression of the pre-B cell receptor. This initial form of membrane IG activates several rounds of pre-B cell division, followed by the rearrangement of the light chain V and J gene segments, allowing the cell to express mIgM. It is now an immature B cell and leaves the bone marrow. Once the completed IgM is expressed on the cell surface, the immature B cell leaves the bone marrow to complete its maturation through the transitional T1 and T2 stages in the spleen. As T1 transitional immature B cells, they enter the splenic marginal sinuses, percolating into the T cell zones. Here the immature cells undergo positive and negative selection. Those that avoid negative selection differentiate into T2 transitional B cells, which gain the ability to enter the B cell follicles, where they complete their differentiation into mature follicular B cells and recirculate in the blood. Mature B cells recirculate between the blood and the lymphoid organs, entering the B cell follicles in the lymph nodes and spleen, where they respond to antigen encounter in the presence of T cell help by generating antibody responses.